evening, students. Yes, Shillong. Good evening. How are you? Yes. Class 11th Biology, Shillong. Just wave me your hands if this you. Just wave. Just. If this is your class, please. Okay, fine. Very good. How are you? Everybody at Shillong, how are you? Yes. Many are back from the school. Okay. After a hectic day, I guess. Okay, fine. So, how are your studies going on? How are your studies going on? Yes. How are your studies going on, Shillong? So apart from Shillong, any other centers logged in? Just let me know, please. Apart from Shillong, just ping me with a good evening or hello, hi, whatever. Okay, I guess now the centers are okay, fine. So, how is biology going on? How is biology going on? Yes. How is biology going on? Good. Everything is good. Nice. Nice to know this. Very good. So, what was the last thing we were talking about in biology? What was my last class about? Yes, Shillong. What was our last class about? What were we talking about? Yes. Tell me, what was the last class about? We finished a chapter, isn't it? We completed a chapter in the last class. So, did you start solving questions from that chapter? Did you? Did you start solving questions? Tell me. Yes, children. How many of you have started? Just raise your hands. How many of you have started solving questions from the chapter? You have started reading the chapter. You have a test also this week. Digestion and absorption. Please do appear for the test. Every center, each and every center, all of you. That's important to know how your studies are going on, so that I can put in more efforts if it is not going on in that way as expected. Actually make you put in more effort. Okay. Fine. Yes. Raise your hands. How many of you have started studying digestion and absorption? Please. Raise your hands. No, nobody. Nobody has started studying digestion and absorption. Why? You have not started studying digestion chapter? Why? The reason being? Are you planning to bunk the test? Yes, Shillong, children? Tell me. Answer me quickly. Yes, how many of you have started? Nobody. Please do start studying the chapters as soon as they are completed over here. That will reduce your burden for the last moment, isn't it? If you keep on things pending, that is going to be very, very painful at the end of the day. Okay? Everybody, please. Just keep in mind, you have to study everything from now onwards itself. Don't take things very lightly. But yes, be regular. Be regular with whatever is being taught in the class. So we are supposed to start a new chapter today. Is it? What's the chapter? Which chapter? Yes, Shillong, tell me. Shall I continue with human physiology? MCQs? You want MCQs? You want to have MCQs? What? What MCQs? Your test, yes, your test is MCQ based. The test will be MCQs. Total, you have to give your test in MCQs, multiple choice question. Remember one thing, there will be negative marking in the test also. So don't do any kind of guessings. If you're sure about an option, then only go for it. Otherwise, you will lose marks also. If you get 4 marks awarded for a correct answer, -1 will be for an incorrect answer. So keep that in mind. Okay? Don't go for random marking. Just whatever option I feel like, I will go ahead. Because you have a negative marking. Negative marking does affect your score very, very badly. Just keep that thing in mind. It does affect your score very badly. So please, everybody, all of you. Okay? Yes. You want to have MCQ questions? Okay, fine. We will do some MCQs from the digestion chapter. From which chapter do you wish to have MCQs? Tell me, children. Tell me, please. From which chapter? 2. What is 2? What is 2? Write the name of the chapter, please. Okay, fine. So let's do some questions from breathing and exchange of gases. Okay. Okay, fine. Okay. Okay. So look over here. Have I made you solve these questions? Did we solve these questions earlier? Just let me know. Have you solved these questions earlier also? Yes. Have you solved these questions earlier? Yeah, breathing was finished in last class. Let me know if you solved these questions earlier. Yes, children. Yes, I can see one more center logged in. Okay, St. Joseph's, good evening. How are you? Yes. Tell me the correct option. Shillong. St. Joseph, you were not present in this class. You need not worry for now. Shillong wanted to have some MCQs from the previous chapter. We can just wait for a few minutes. We'll start with the new chapter, in a moment. Okay, fine. Very good, Shillong. Nice. Okay. Tell me this. Just bear with me for 10 minutes. Within 10 minutes, we will start a new chapter. Okay. Did we do this question? Opening is covered by glottis. Yes, the opening of glottis is covered by epiglottis. Very good. Okay. This question, wish to answer this question. Instrument that measures the amount of air inhaled and exhaled with each breath. No. Sphygmomanometer is for measuring barometric pressure. So if you wish to measure the pressure at Shillong, yes. If you wish to measure the pressure at Shillong, that what is the particular pressure, environmental pressure at Shillong, you will be using sphygmomanometer. If you want to measure my blood pressure, you will be using sphygmomanometer. So sphygmomanometer is an instrument to measure blood pressure, pressure measurement. Okay? And spirometer is an instrument which measures the particular volume of air during each breath, expelled, inhaled, whatever pressures we have studied till now, all that will be measured using spirometer. Okay? Fine? Okay. Answer me this question. This one please. Amount of air in the lungs that remains after forceful expiration. What is that particular amount of air called? Excuse me. What is the amount of air called which is remaining in the lungs after forceful expiration also? Yes. Quick. Shillong, fast. Be fast. It's an easy question. Exactly. It's called as residual volume. Very good. Okay. This question. Hemoglobin retains oxygen and releases it in the where? Heart, lungs, glottis, tissues. Where is the oxygen released by hemoglobin? Very good. You're doing nice. This question. Yes, children. Wish to solve this question? Yes. Bulk of CO2 released from the body tissues into the blood is present as, what's the form? 70% carb amino hemoglobin, 30% bicarbonate carb amino hemoglobin in RBC, bicarbonate in blood plasma and RBC, whether it is free CO2 in the blood plasma. What is the bulk? Quickly. This answer, even St. Joseph's can try. This was the thing I taught you in the last class. Yes, St. Joseph, you can try this question. This was done in last class that we did on the last Tuesday, the last day of the chapter, this chapter with me. So you can try this question. This is easy for you. We have done this thing. Please try. Shillong. Please try. No? St. Joseph? No? Okay, you are telling me A option, is it? Shillong? Yes. Are we connected, Shillong? Okay, fine. What's the option for this? Correct option for this question? Yes, children? Correct option for this question. Come on. Let's see. Okay. CO2 transport. CO2 transport was done by 3 means. Remember? The first was dissolved in plasma. How much it was? It was 3%. Then we had next particular part, CO2 transport, that was what? That was in the form of bicarbonate. This was 70%. And then just refer to your notes in from the last class. We have done this question. This should have been done by you easily. This is carb amino hemoglobin. And how much was this? This was remaining 23%. So obviously the majority of CO2 is transported by bicarbonate form. So this is the bulk transport medium. So the correct option for this question would be what? The correct option for this question would be bicarbonate in blood plasma and RBC. This would be the third option. Please note down this question. This is a good question. Both the centers. Please note down this question. Note down this question. Charger. Yes. Note down this question. Did you? All of you. St. Joseph. Shillong. Noted, children? Is this clear to one and all? Okay, just bear with me for 2 minutes. My laptop has shut down because of no charge. Okay. So in the meantime, let's talk about the bicarbonate transport. So what was bicarbonate transport? What do you understand by bicarbonate transport? What do you understand by bicarbonate transport? Tell me, children. Shillong. Yes. St. Joseph. What was bicarbonate? What was the enzyme required for bicarbonate transport? Let's see who is going to answer first. Is it Shillong or St. Joseph? Let's see who is going to answer me first. Quickly. Yes. Name the enzyme required in bicarbonate transport. Quick. Shillong. St. Joseph. Yes, children. Okay, I guess I need to... Okay, good evening, Testo College. Yes, Shillong, answer please. St. Joseph. Name the enzyme that participates or that makes the bicarbonate ion. Exactly. And St. Joseph, what do you think? The enzyme? Name the enzyme that does this particular thing. Good evening. Name the enzyme. Remember CO2 plus H2O, that is by the means of an enzyme carbonic anhydrase converted into H2CO3. Okay, this was reversible, sorry. Converted again by carbonic anhydrase. Into what? Into H plus and SCO3 minus. So this was the bicarbonate section, isn't it? So very good. Keep it up, Shillong. Very nice. Okay. So let us start a new chapter now. Okay, we will do some more questions in the other classes. For now, let me start a new chapter. So what was my chapter? What I was supposed to start teaching you today? It was body fluids and its circulation. Okay. Let's start with this chapter. Again, one new chapter from the human physiology section. Okay, so what are body fluids? What are body fluids? Who's going to tell me? What are body fluids? Yes. Texo College, Shillong, St. Joseph. Yes. Fluids. See, it is body fluids. That means it needs to be more than one. What is the other body fluid present in your body apart from blood? Yes, children. What are the other body fluids present in your body apart from blood? Yes, name them. Quick. See, you have two body fluids present in your body. One is blood and the second is lymph. This is a new chapter, so all of us can start with a new beginning. Yes, platelets and plasma are not body fluids, children. Plasma, in fact, is the watery part of your blood. We will talk about this. But lymph is correct. Very good. Lymph is correct. So we have two body fluids, one is blood and other is lymph. So let me discuss blood first and then I will discuss the lymph section. So when you talk about blood, what do you think about blood? First of all, let us be very, very clear that there are two types of circulatory system. There are two types of circulatory system. One is said to be open, and the other is said to be closed. So we have two types of circulatory system. What are they? Number one is open, number two is closed. So open circulation and closed circulation are the two circulatory systems you will find across the animal kingdom. So whenever you talk about circulatory system, we will talk about either closed or open. So what kind of circulatory system do you have? What do you think? It's closed or open? Yes. Tetsu College, what do you think? What is the particular type of circulatory system do you have? Yes, Shillong, very good. Yes, St. Joseph, what do you think? Tell me, children. St. Joseph, what do you think? It should be closed or open? What are you having? A closed circulation or open circulation? Yes. Answer, please answer. There is no negative marking for answering now. Obviously in test there will be negative marking, but now there is no negative marking. You can answer freely. Neither I can come to you people also. Yes. St. Joseph, Tetsu, please answer. What is the typical type of circulation you have? Yes, tell me quickly. So let us draw both the things and then you will tell us what is your type. Okay, fine. So what happens in open circulation is basically there will be a big pipe. Okay, a big pipe will come at your main door and the pipe will be containing water and it will be rushing through all your rooms. Your kitchen, your washrooms, bedrooms. So all your rooms will be having water access through the one big pipeline. This is open type of circulation. Okay, this is a big vessel. And this is open space. Called as lacunae. So lacunae in biology is always an open space, a space which is not closed by any kind of boundary. Then in closed what happens is your body is actually having certain branching system. You have an artery divides into arterioles divide into capillaries. The capillaries they again reunite to form venules, and the venules they form big veins. So this is artery, this is an arteriole, this is capillary, this is venule, and this is vein. Is it? This is what you were talking about, closed circulatory system. So if you are going to supply the things to tissues, that will be only and only through capillaries. See, there is no direct access of the blood to the tissue. The tissues are not bathing in the blood. Whereas in the open circulatory system, you are actually making the tissues to bath in the blood. See, this is a tissue. Let's mark them in orange. I have many colors, so I have a very good option. Okay, so what is happening over here? Oh, sorry. So what is happening over here? You are taking tissues and you are bathing your tissues into blood. That thing is blood. Okay. So what are you having now? Any idea? You have open, you have closed. Yes. St. Joseph? What do you think? What are you having now? What can you say? You surely have a system which is very, very typically arranged, and that is said to be what? That is said to be the system of closed circulation. So you have everything arranged in a very, very sequential manner. That is, you have arteries dividing into arterioles into capillaries, and capillaries from which the tissues can derive their things to be used, like gases, food, everything. And then capillaries, they reunite to form venules. Venules reunite to form veins. So all these structures are showing a level of evolution. So obviously, you are having what? You are having a closed circulatory system. I will give you an example where you have an open circulatory system. Example in cockroach. Okay. So cockroach belongs to, you must have studied this thing in junior classes. There is a phylum called as Arthropoda, the largest phylum on the earth. You are going to study this in chapter number 4 also, animal kingdom also. So this cockroach or Arthropoda is the largest phylum on the earth, and they have what kind of circulation? They have open circulation. And you have two examples from this. You have Annelida and Chordata. Annelida is the phylum having earthworm. Okay, we all have seen earthworm. Especially during rainy seasons, they are in abundance. So we all have seen earthworms, especially in rainy seasons, and the earthworms, they have what kind of circulation? They have closed circulation. And obviously Chordata, me and you. Tiger, cow, frog, fish, crocodile, wall lizard, they have what kind of circulation? They have closed circulation. So obviously closed circulation is more evolved compared to open circulation. So this is what is the pathway of blood taking place in your body. You have what kind of circulation? You have closed circulation. Okay, that means the blood is flowing in vessels with proper branching. Clear? Everybody? Shillong, are we connected? St. Joseph, Tetso College, you people, all of you are quiet. I guess we are not connected or what? St. Joseph, Tetso, are we connected? Just let me know if it's clear or not. Children, how would I know if we are connected or not, or you are getting or not? If you are understanding everything, just let me know, please. Yes, Shillong, you can give me a thumbs up, since I can see you people. Yes. Clear? Everybody? All of you, just give me a thumbs up. Everybody. Yes, Shillong. Oh. Yes. Did I miss your thumbs up? There was no connection signal visible to me. Did I miss your thumbs up? Can you just repeat? Rewind your thumbs up, please. Shillong. Clear? Clear with one and all? Quickly give me a thumbs up. Okay. Very good. So let me proceed further. Nice. Very nice. Okay, let me proceed further with the composition of the blood. Now, when you talk about closed circulation, when you talk about open circulation, things are clear in your mind that yes, you are having a proper system of channels where you will be having vessels, you will be having pipeline to deliver water in your kitchen, water in your washroom, isn't it? Water in your garden. So every part will be segregated. And in open, what happens is water is coming directly at the main door and it is being gushed to all parts of the house without any specificity. So what is more evolved? What is better? To have an open circulation or closed circulation? What is better? What do you think? Which is better for you? Which one of these is better for you? Testo? St. Joseph? Yes, exactly. You have your closed circulatory system constructed in a way that there is more precision, isn't it? So closed circulation is always better than open circulatory system. Better instead of better, I will be writing more evolved, isn't it? Clear? That's so. Are we connected? Tell me, children. Both of you. Are we connected, Tetso and St. Joseph? Yes. Are you getting whatever is being taught? Tetso College? St. Joseph, I guess, has lost connection or is an issue. Are you getting whatever is being taught, Tetso? Okay, fine. So, we have next topic, that is the composition of blood. So, blood is basically composed of two components. Let's see, what are they? The first component happens to be the plasma, and second component happens to be the formed elements. Okay? Now, while plasma is having 55% of the total blood volume and formed elements only 45%. Okay, don't be sorry for that. That's so, it's okay, I understand. Okay. So, you are getting everything. So, this is the composition of blood I have started with. This is the composition of blood. Okay. Fine. St. Joseph also, I guess, is not having connection properly. Today, Kohima is also not there. Okay. Okay, fine. So, plasma. Let's talk about plasma. What is plasma? If a question comes to you that what is plasma, you will write that plasma is the watery component of the blood. Next thing what you are going to write, it is rich in, it is yellowish in color, okay, and yellowish means what? It is straw yellow. Light yellow in color, that means. Then you have the components. So what is the main components? Let's see the components of plasma. So your components of plasma, you have water first of all. Second you have, after water you have certain electrolytes. Or what you say in general terms, ions. What are ions? Your ions are sodium, potassium, calcium, iron, magnesium, manganese, and many more. There are many ions. Okay? Next, what do we have in plasma? We have certain proteins. What kind of proteins do you have in plasma? The first and foremost, we have a particular protein called as albumin. The role of albumin, what is the role? It maintains osmosis. Then you have next protein called as globulin. What is globulin doing? It helps you in defense, protection, immunity. Next is fibrinogen. What is fibrinogen doing? It is clotting, helpful in clotting. We will see this particular things. Okay? Now apart from these components, you have glucose, you have fats. Okay? So you have all these components present in your plasma. Okay. What is osmosis? What is osmosis? Yes, children? What is osmosis? Answer me. Otherwise I will color a whole of the notes in yellow. Quickly answer me. I want answer from each center. I will keep on doing this thing unless every center is answering me. See, I will keep on doing this thing. Answer please. Yes, Shillong. Yes, good, but not correct. I will just rectify this thing. Yes, it is right. Okay, it's right. It's not wrong, but I need more of content. Just try to put more of content. Yes. Just try once more, Shillong. You can answer. It's correct. But from where to where? Is it correct? It is diffusion of water. Very good, Tetsu College. It is diffusion of water from where to where? Okay, let's solve a question. Let's see from where to where. Did you copy everything? Did you copy everything till now? We'll see. Both these definitions given by both the centers, Shillong and Testo. I am missing a small component in both of them. We'll see what component. You will answer this question. Testo, copy it. Yes, Saint Joseph, where were you lost? Where were you, children? Connection problem. Okay. So let's start with a question. Okay, I'm just, yes, very good. Yes, Testo. See, this is A fraction, this is B fraction. Now, what I am doing is, see Shillong, for osmosis we always need a semi-permeable membrane. That is why I have made a dotted structure to show the semi-permeability of this membrane. Exactly. So this is my solution A, this is my solution B. We are going to study osmosis. Okay, now this solution A has, are you studying molarity in chemistry? Have you completed with mole concept part? Are you aware about normality, molarity terms? Just let me know. Have you completed molarity, all those terms in chemistry? Okay, no issues, let it be, don't get tensed. Now, just imagine we have a solution. Okay, you have completed, so I can give you on that ways. This is a 30 millimolar or 30 molar solution of Na+. The molarity of A solution is 30 molar. Okay, and the water, and this molarity is how much? This is 10 molar. Na plus. So what you are having is, you are having 250 ml of water over here, and you have again 250 ml of water over here. So you are having 250 ml of water on either sides, and you are having what? You are having 30 molar solution of sodium on one side and 30 molar solution of sodium on other side. Clear? Clear with the question? Now you have to tell us that movement of which of the following statements is correct. The first statement is, what is going to happen? I mean to say, movement of sodium from A to B. Second is movement of sodium from B to A. Third is movement of water from A to B. And fourth happens to be what? Movement of water from B to A. So what do you think? What will be the correct option? Yes. Yes, Shillong? St. Joseph? Tetso? Answer this question, please. This is A, this is B. I'm just remarking it. Okay, start. Start solving this question. Yes. Tetso, your answer? You are the only one left to answer. Please answer. Yes, I've got answers from, in fact, it is both of you have answered the same option. Yes, Tetso, your option, please. Your definition was very much correct. Shillong, you're changing your options? Changing your options. This is final? Okay. Is this final from Shillong side? Yes, Shillong children. Is this final? Again, you're changing your option. Which one is correct? What is it? Final answer. Final. Give me a final option. A is final. Okay, fine. Sure? Are you sure about it? Okay, fine. So you have, like, in fact, answered all the options on the available list. Okay, fine. So, definition, first of all, I will write the definition of osmosis and then we'll try to solve the question. Okay, so I'll just erase this thing from here. Make some space. Yes. Osmosis. Let's see. Movement of water molecules from its higher concentration to its what? To its lower concentration, and through what? through a semi-permeable membrane. So what is the keyword? Let's quickly mark the keywords of this. So its higher concentration to its lower concentration. Keywords. And movement of what? Movement of water molecules, isn't it? Movement of water molecules. So, let's see which of the options. So, obviously in the solution A and B, only the movement part will be courtesy what? Will be water. Never the sodium is going to, since sodium is the solute part, sodium is never going to move. So this property of osmosis is only for solvents. Okay? So if we talk about osmosis, it is only for solvent, but never for solute. Keep this thing in mind. Okay? Clear? So this clearly rules out the first two options. Why? Because they were talking about sodium movement. Whereas according to definition of osmosis, sodium is not the component moving. What is moving? Water is moving. So it could be either C or D. Let's see. So movement of water from its higher concentration. So there is more of water available here. Why? Because there is less of ions. If there is less of ion, that means there is more free water molecule. Isn't it? If I engage all of you with a book, all of you will be busy. But if I am just giving 2 books to somebody, rest of you all will be free. That means you have more free water molecules when you have lesser number of books. Isn't it? Or more free students when you have lesser number of books. So there is more book over here, 30 molar, and less books, that is 10 molar. So there will be more free students here. That means free water will be higher on this side. Clear? This is the higher volume of water. Isn't it, children? Are you getting my point, everybody? Each and everybody. Tetso, St. Joseph, Shillong. Are you getting my point? You must be. So you have 10 molarity of sodium. That means there is more of free water here and less of free water on this side. So water is going to migrate from which side to which side? Yes. Tell me. Water will be moving from which side to which side? From this to this side. And it will keep on moving until this particular thing is equal to this particular thing. Because you have done mole concept, you are aware about the things, so you know that it will be done till the point both will be equal. So we started with 30, 10, but osmosis will make both of them equal. So finally you will have water spread across in a way that there will be $50 molar solution over here and 50 molar solution will over on the other side. So this is what is called as osmosis. So according to this, the migration will be from where? It will be from B to A. None of you answered this option. All of you answered, but not this. Okay, no problem. Is it clear now, Shillong? St. Joseph? Tetso? Is it clear now? Will you make more mistakes now? Okay. Fine? So how many of us know that patients with high BP are not allowed to have extra salt and sugar? How many of us are aware about that thing? Yes. Shillong can just show me their hands up. Yes. Listen, you just give me a hands up. Show me a hands up. Yes, Tetso College, you need to type. St. Joseph, please type. Yes. How many of you know that we don't allow people with high BP to have more of salt and sugar. If there is a patient of high blood pressure, you will not allow them to have more salt and sugar. So if you become a doctor, you are not going to allow them. Why? If some person is having a high blood pressure, okay, high blood pressure means what? High blood pressure means there is more of water in the plasma. Because of more water, what happens is let me draw again, there is more of water, first thing, then because of more of water, there will be more blood volume, and because of more blood volume, what is going to happen? There is going to be high BP. Now suppose this person is having more of sodium, isn't it? So from intestine the ions are absorbed. This is absorption. This is not osmosis. This is what? This is absorption. Okay. It can be passive or it can be active. You know what is passive and active. Passive is no energy and active is energy required. So if you start transporting sodium ions into this, a time will come, then there will be high sodium also. And when there is high sodium, it will be always followed with water. Why? Because water moves from its higher concentration to its lower concentration. So if this is more of sodium inside, if sodium volume increases inside, what is going to happen? Water is also going to increase inside. Gradually the blood volume will also increase if water increases, and the blood pressure will also increase. That is the reason why we don't allow people with high blood pressure, already high blood pressure, to have more sodium or more sugar. Both of them are harmful for them. Why? Because this sodium is going to enter and this sodium is going to attract more water. More water means what? More blood volume, and more blood volume means higher will be the BP. Clear? Shillong, Tetso, St. Joseph, clear children? Okay. So this osmosis concept is clear, isn't it? Okay, very good. Okay, let's see the formed element section. Copied everything? Okay, let's start with the formed element section. So what do you have in formed elements? Yes. You have three types of formed elements. One is RBC, WBC, and platelets. We will deal with all of them one by one. So you have three formed elements. What are they? RBC, WBC, and platelets. Yes. So we'll start with RBC. Okay. You want to have a break before we start with RBC? Wish to have a break or shall I continue? Yes. Okay, then have a 5 minutes break. Quickly after 5 minutes we will resume with RBC, red blood cell or erythrocytes. Okay, fine. Okay, children, let's resume our class. Yes, everybody is back from break. Shillong, Tetso, St. Joseph, back. All of you. Yes, Shillong. Okay, fine. So we are talking about RBC, called as red blood cells or erythrocytes, isn't it? So first and foremost thing, what comes in our mind from the last chapter, obviously, we have a particular knowledge that yes, RBCs are the particular cells that have a color, isn't it? Color, red colored cells. Why? Because of a particular pigment called as hemoglobin. So let's talk about this thing. First of all, we will talk about number of RBC. So it's 5 lakhs per mm cube of blood. When we talk about males, and it is 4.5 lakh per mm cube of blood in case of females. So you have two numbers, 5 lakhs in male per mm cube of blood, and it is 4.5 lakh per mm cube of blood in case of females. So it has a particular pigment called as hemoglobin that gives what? That gives red color. Fine? Next thing, what is the function? Yes, what is the function of hemoglobin? What is hemoglobin doing? Quickly. This carries oxygen, isn't it? This carries oxygen from the alveoli to your tissues. So, for this function, I would say the RBC is so very dedicated that RBC has no cell organelles. Ma'am was teaching you cell organelles, I know. What are the various types of cell organelles ma'am has taught you? Tell me. Yes. Name a few cell organelles that ma'am has taught you. Yes, children. You are studying cell in the botany section. What has ma'am told you about cell organelles? Anybody among you? Yes, very good, Shillong. Tetso, what do you think? What are cell organelles? The small organs of the cells are called as cell organelles, isn't it? Mitochondria, chloroplast, Golgi, lysosome, endoplasmic reticulum, vacuoles, glyoxysomes, peroxisomes, and many more. All these are what? These are the cell organelles. So your RBC is very, very special. Why? Because every cell has cell organelle, but RBC has no cell organelles. Mind it. And all the cells have nucleus, but RBC has no nucleus either. So they don't have what? They don't have cell organelles, they don't have nucleus. Why? Why are they eliminating out the things? Why? To accommodate maximum amount of, I'll just write HB for hemoglobin. So to accommodate maximum amount of hemoglobin, what your RBC is doing? Your RBC is removing everything from its interior. It's just making space for only and only hemoglobin. And to do what? To carry more amount of oxygen. So if it modifies itself in a way that it is removing everything, it is very, very specific in its goal that I have to accommodate what? I have to accommodate hemoglobin and I have to transport oxygen. Very, very clear in its goal. Crystal clear. Isn't it? Clear? Let's talk about its shape. The shape of the RBC. RBC is biconcave in structure. And disc-like. Better I would add this thing over here only. Biconcave and disc-like together. Disc-like, comma, biconcave in shape. So we did do this thing in the last chapter. This was something I was talking about. The typical shape of your RBC. It's biconcave in nature, disc-like. So it's just like a disc frisbee. As you have seen frisbee, just circular in nature. But yes, the frisbee will be towards its inner side, it will be having a bend, like this kind of thing. And from the lower side, bend towards outer side. So this frisbee will be thick on the edge, but thin on the center. Okay? Clear? Fine. So, I don't have a clear diagram of RBC. Okay, fine. Clear? Are we done up to this? Everybody? Clear with Tetso, Shillong, St. Joseph? Tell me, children. Is everybody clear with this thing? Shape of RBC. RBC is biconcave in shape, disc-like in structure. Now, moving ahead. Where is RBC formed? RBC is formed in bone marrow. So, your long bones, they are having certain structure inside them. They're not hollow. The birds have their bones hollow, but the long bones of the long bones of mammals, they're actually filled with some marrow-like structure. Marrow means a filling. It's called as bone marrow. And this bone marrow is the site where RBC is formed. Red blood cells are formed. Okay? So after their formation, they do their activity. They transport oxygen. They do whatever roles they are being assorted. We have seen those roles in the respiration section. Isn't it? We have done the chapter very nicely. And when it moves away from the bone marrow, what is that? Particular RBC has a lifespan. Has a lifespan of how many days? 120 days. So what is going to happen if RBC completes its 120 days? After 120 days, RBC goes into what part of your body? Into spleen part of your body. Okay? Now what is a spleen? How many of you have heard this word earlier also? Yes? No, it's not liver actually. Shillong, it's just a small pink organ present just below your liver. Okay? So once the RBC completes its lifespan of 120 days, it goes into an organ called as spleen, where it is destructed. So can we call spleen the graveyard of RBC? Can we call spleen to be graveyard of RBC? Can we? Yes, we can. We can call it to be a graveyard of RBC very much. Clear? Clear with this, children? St. Joseph. So what is spleen? This is spleen. Small pink organ present below the stomach. Okay. Now we were talking about that RBC does not have nucleus, isn't it? RBC does not have nucleus. This is the case with mammals. Just rectify this once again. Mammals do not have nucleus in their RBC, but mammals do not have nucleus in their RBC, but we have exceptions, except camel and llama. LLAMA. So camel and llama are the two animals which are mammals, no doubt, which are mammals, but they don't have enucleated RBC. So we can write down mammals have enucleated RBC. What is enucleated? Means without nucleus. And camel and llama are what? They have nucleated RBCs. I'll just send you a pic of llama in your group. You can just refer to the llama. How does it look like? Okay, I'm not connected to Testro. Testro is Dimapur, isn't it? But I don't have St. Joseph group added. Okay, no problem. I will ask somebody to add me and I will be sending that in the evening. Okay, so llama is basically an animal which looks like a hybrid of, those people who have got the image, they can see, it looks like a hybrid of camel and sheep. So it looks like the camel is having the whole fur-like appearance, but a height of sheep. So it's an animal which is basically found in cold regions. Okay. Fine. Clear up to this? So your nucleus, you have a RBC which is having no nucleus. But camel and llama are having an RBC with nucleus. They have nucleated RBC. Okay. So can we do a question? Things are clear up to this? Everybody done with the RBC section? Is everything clear? Shillong, St. Joseph, children at Testo College, let me know please if things are clear. Okay. St. Joseph, I'll be sending you those images in the evening. I will ask somebody to add me in your group. I'm not over there. Okay, I'll just ask somebody to add me and then I will be sending you those images. Or you can also ask your coordinator to add me on my behalf. That would be much faster, I guess, because you people are indirectly contact with those people. Okay, so let's have a question. I'll give you 5 options. Yes. Solve this question. St. Joseph, Shillong, Tetso, please try and solve this question. Yes, Shillong. Yes, children. Quickly sort this answer. Sort it out. Quick. Shillong, Tetso. I've got an answer from St. Joseph. And still waiting for Shillong to reply. Shillong and Tetso. Please reply. Yes, children. Are we connected? Okay, let's sort this out. I guess we are not connected. So let's sort it out. So RBC is nucleated. Okay, I told you. Yes, Tetso. Good, nice, very good. So I told you that RBC is not having nucleus. Okay. I always told you don't be in a hurry to answer MCQs. Analyze the question first. Look over, give equal importance to all the options. So I told you one statement over here that, see, I told you what? I told you the mammals, isn't it? I told you mammals have E-nucleated RBC. That means a mammal will not have a nucleus in their RBC. Then I told you about an exception also. See, these 2 exceptions, camel and llama. So obviously camel and llama, they have nucleus in their RBC. So this is correct. Both of them are correct. Frog is not a mammal. Okay, frog is not a mammal. Frog is an amphibia. So, frog will also have a nucleus in the RBC. And keeping this in consideration, you have to give an answer that satisfies all the options, that is all the above. So, the correct option for this question would be all of these. So, it's a fish, it's a frog, it's a wall lizard, crocodile. They will all have nucleus in their RBC, because without nucleus, RBC is a character of what? Which particular class? It is characteristic of mammalia, not of every class. Okay? Fine? Clear with this? So fishes, amphibia, reptiles, aves, all will have nucleus in their RBC, but not we. We are mammals. Okay, fine. So this is all about your RBC. All this is about your RBC. Let me start with WBC section. So white blood cells, they are a big group of cells which are actually grouped further into two subcategories. Let's see what is the subcategory. Now first of all, its number is 5000 in 1 mm cube of blood. Number of WBC. We will divide it into the ones with the granules and the other without granules. With the granules, they are said to be what? They are said to be granulocytes. Granulocytes means what? With granules. And the ones without granules are said to be what? They are said to be agranulocytes, means no granules present. Granules are the small grain-like structures which are present inside the cell. They can be present in granulocyte, they cannot be present in the agranulocyte. WBCs are the only category of cells which are having these particular properties. Not every cell is having granules. Okay. Now, if you are having granules, you will be having again three different types of cells under granulocytes. Let's see what are they. The first is neutrophils. The second we have is eosinophils, and the third we have is basophils. These basophils are also called as mast cells, and eosinophils are also called as acidophils. Okay, and agranulocyte, I will just take it to the next page. Okay. The agranulocytes are further divided into two types. What are they? The first is called as monocytes, and the second is said to be what? It is said to be lymphocyte. So we have different categories of cells. We have monocytes, we have neutrophils, we have eosinophils, we have acidophils, basophils, mast cells. These are different types of cells we are having. So we have neutrophils, we have eosinophils, we have basophils, we have mast cells, we have monocytes, and we have what? We have lymphocytes. Clear? Yes, Shillong, I cannot see you. People are stuck. Just let me answer. If not on the chat box, if chat box is not working, please answer me on the WhatsApp group. I need to know if you are getting things or not. Please answer me over here. If you can, if you are able to attend the class, answer me on the group. If your chat box is not working. Yes. Tetso, St. Joseph, are you people clear? I guess we are connected. Are you people clear up to this? The types of WBC, let me know. We have 5000 mm³ of WBC present in our blood, per mm³ of blood, in fact not total body but only in 1 mm³ of blood. And you have two types of WBC, that is granulocytes. Granulocytes, let's look over their numbers. You have granulocytes 72%, granulocytes only 28%. Okay? Then you have is neutrophils. Neutrophils are round about 60 to 65%. You have eosinophils, that is 2 to 3%. You have basophils 0.5 to 1%. You have monocytes 6 to 8%. And you have lymphocytes roughly in between 20 to 23%. Okay, so this makes your total WBC's composition. If you just closely look at this, your neutrophils are the highest in number and basophils are the lowest in number. Okay, let's look over each and every cell's property individually. Let's start with neutrophils. Shall I start? Yes, Shillong. I guess you don't have phones also now. Really sorry. Okay, Tetso, St. Joseph, can I proceed ahead? Is it clear up till now? Okay, so let's talk about neutrophils. What I will do is that I will take all the granulocytes at one go. So in one go and the agranulocytes in one go. So neutrophils, we had eosinophils and we had what? We had basophils. So we'll do these three cells in one go. That is all the granulocyte section. So phil means loving, phobic means fearing. So if it is neutrophil, that means it must be loving something which is neutral in nature. The question is what? If it is eosinophils, it must be loving something which is, I told you one more name for this, acidophils, which is showing its love towards acids. Okay, and you are having basophils or mast cells that shows its love towards something which is basic. Now, I told you in first place only that these are the cells which are having granules and these granules can be stained. So based on the staining properties, what we have is these are stained by acidic dyes like eosine red. So eosine red is an acidic dye which can stain eosinophil. That gives its name eosine phil or acidophil because this is an acidic dye. This is stained by basic dyes. I'll give you an example like methylene blue. Okay, so you have an acidic dye, eosine red. You have a basic dye, methylene blue, which is staining these cells. Clear? And what is neutrophil? Tell me what is neutrophil? Neutrophils are stained by both acidic as well as basic dyes. So if you're talking about both acidic and basic dyes, you'll be talking about neutrophil. If you're talking about only acidic dyes, you'll be talking about basophils. Acidophils. So acidophils, oh, this color is not working. Sorry. We'll have this color yellow. This works everywhere. Yes. Acidophils stained by acidic dyes. Basophils stained by basic dyes. And stained by both acidic and basic is the neutral one, neutrophil. Clear? Copied up to this? Everybody? Okay. Fine. Shillong is not connected. There's a problem at Shillong Center for internet connection. Okay, Shillong people, if you can listen to me, you can just WhatsApp me on the group if you have your phones available with you. Okay? Okay, fine, St. Joseph, very good. So, neutrophils, they are the highest in number. Next property about neutrophil is what? They are highest in number. And they perform a specific function called as phagocytosis. What is phagocytosis? Phagocytosis means cell eating. So when a cell is eating, it is said to be what? It is said to be phagocytosis. You must have heard this particular thing when you are doing the particular functions, when we are discussing the functions of amoeba. Amoeba also performs phagocytosis. So this is what? This is a cell. Okay, this is its nucleus. Since they are nucleated, the WBC is nucleated. Now it sees a particular particle outside it. What happens is, it starts moving towards this particle and make its own space. Let's have this particle in other color to make it more appealing. Yes, it starts to move towards it, and gradually what happens is, after this step, they engulf it. This was the particle in magenta. Yes. And this process is called as phagocytosis. This whole process is called as phagocytosis. Once it engulfs it, once it is engulfing it, what happens is, it breaks it into small pieces and then throws it out. How? How does it throw it out? It throws it out by the process of, again, it is by process of reverse phagocytosis called as exocytosis. So if it is endocytosis, the cell is taking into water or some soil substance. And if it is exocytosis, the cell is actually throwing out the substance outside. So this is what is endo and exocytosis, phagocytosis on a whole. Okay, clear? So these are the typical cells that perform phagocytosis. So obviously, what is it doing? It is going across your body, moving across your body and trying to get out the culprits. So it is going to attack those things and it is going to destroy them. Okay. Now this is eosine red. What is this? Oh, sorry. This is eosinophil. What is this going to do? This is basically, this number of these cells, they increase in allergy. Any kind of allergy. Better, I would say, increases in parasitic infection other than allergy. I would say increases in parasitic infection. So if there is a parasite in anybody's body, the parasite can be anything. It can be a bacteria, it can be a virus, it can be a worm, it can be anything. So if there is any parasitic infection in the body, this number of this eosinophil, they increase automatically. The parasite can be anything. Now, what is this doing in your body? This is the one which is allergy mediator. Mediator means it helps in causing allergy. This causes allergy. Because of a chemical called histamine. So what is histamine? Histamine is a chemical which is present, histamine is a chemical which is present inside the basophils. So these histamines, when they come out from the basophil, what they do is that they cause allergy. Clear? Clear with everybody? Just let me know, please. The functions, I will move ahead. If not, I will explain again. Yes, children? Clear with one and all? Tetso, St. Joseph, Shillong. Tetso, St. Joseph, Shillong. Yes, clear? Okay. Can I move ahead? Did you copy everything? Please reply. St. Joseph, Tetso. Okay, please copy. Cause of allergy is histamine. Histamine is a chemical which is present inside the basophil. Okay, now when there is a particular environment, these basophils, they blast. They just open up, and what they release is, they release histamine. Histamine is a particular component which causes allergy. Okay, so whatever allergy symptoms you are having, they are basically because of this histamine. So if tomorrow you become a doctor and a patient of allergy comes to you, you will always give them a drug to decrease the histamine content. It's called as antihistamine. The pumps of asthma patients, they contain the drugs which are antihistamine. Okay, in those pumps there are drugs which actually decrease the effect of histamine. Okay, done that so? Clear? Okay, now I'm moving to the next part. That is a granulocyte. Now, what is a granulocyte? The ones without granules, and there are of two types. The one is monocyte and the other is lymphocyte. Okay, monocyte, what is monocyte? Monocyte is very much similar to neutrophil. Why? Because these are also the cells which perform phagocytosis, the characteristic feature. Now, apart from being involved in phagocytosis, just like neutrophils, they are the largest of WBCs. So, if a question comes to you, the largest WBC, you have to write monocyte, and if a question comes to you, highest in number, it would be neutrophils. So neutrophils are highest in number, and you have monocytes, they are the largest in their size. Be very, very clear about these things, okay, don't confuse. So what they do is that they perform phagocytosis, as simple as that. And you are left with only one of them, lymphocytes. They are what? They are the immune cells. And these immune cells are of two types. They are B cells plus T cells. You have two immunity providing cells in your body. They are B cells and T cells, which are responsible for immunity. Okay. So this is the function of each and every type of granulocyte are granulocyte. Their numbers, you are clear with the numbers also. Neutrophils highest with 60 to 65%, followed by you have is lymphocytes, then you have is monocytes and eosinophils, and finally basophils. Okay, clear? Copied everything? Children copied? Clear? Yes, St. Joseph, is it clear? Everybody? Okay. Do a question? Okay. Allergy can be caused by anything. Okay. It can be because of a pollen, it can be because of dust, it can be because of anything. So basically what happens is in under allergy, the mast cells, they burst out. And the mast cell contains a particular chemical called as histamine. And then histamine, what does it do? It starts a particular series of reaction in your body that gradually turns into an allergy symptom. Allergy means running nose, running eyes, continuous sneezing. It can aggravate also. It can become much more worse when the patient actually complains about swelling in the body. It can be accompanied with fever. The patient can, many times they have to rush to the hospital also, and it can be, yes, life-threatening in many cases. So, causes of allergy is different in anyone. So, you might be allergic to some other thing, I might be allergic to any other thing. So, this is basically very, very specific system. Okay. First do this question. Arrange the following in decreasing order. What? You have to arrange these particular cells in decreasing order quickly. We have neutrophils, we have eosinophils. You have to arrange them in decreasing order. Basophils, monocytes, and lymphocytes. Arrange these cells quickly in decreasing order. Largest first or highest first, followed by the lowest. Yes. Start doing it. Done? Type the answer. Type the answer, please. Yes, children. Type it. Clear? Type, please. Shillong is not connected. We have Tetso, we have St. Joseph. Please type in the answer. Yes, children. What is taking so long? In decreasing order, child. You have arranged in increasing order. Just reverse the order. You have to arrange in decreasing order. Okay, the question is decreasing means highest first. Okay, let's see. Let me sort it out. Okay, so you have the highest in number neutrophils, followed by lymphocytes. Tetsu College, but it was a good try. Then you have is monocytes, eosinophils, and lastly basophils. So this kind of question is being asked to learn these particular things. We have certain tricks. Okay, it's okay, it's fine, I got your point. So we have a certain trick to learn this mnemonic, what we call this. You can learn it using neutrophil N, never, L, let, monocyte, monkeys, M, eosinophil, eat, basophil, bananas. So you have to learn this thing. Never let monkeys eat bananas. In that way you can arrange it in decreasing order. So this is the highest and this is the lowest. Clear? This will be an easy process, making you not to refer things always. Okay, you can just learn this thing and you will be able to judge very, very quickly. Clear? St. Joseph, Tetso, Shillong, if connected. Are we clear up to this? Okay. Fine. Percentage composition of this particular chart, Shillong. This thing. Okay, any other questions you want to discuss? Any other thing you want to discuss? Everybody, Tetso, Shillong. Shillong can talk to me on this group. Okay, please copy this, Shillong. First copy this thing, then you will have all the lectures, okay? First copy this thing. Clear? Sorted? Just let me know when you're done with copying, Shillong. We are waiting for you. Okay, I guess maths people are also on the group. They are asking for lectures on relation. Okay, fine. Yes, done? Copied? Biology section, NEET, Shillong. Tetso, we are clear with everything, isn't it? St. Joseph, Tetso. We are clear with everything? Okay? Are we? Okay, nice. Very good. Okay. So can I wrap up the class for today? Can I wrap up the class for today? Tell me, children. Shall I? No doubts? Okay. Fine. Shillong, you copied everything, isn't it? You copied everything? Shillong, children? Okay. Okay, that will be difficult, child. But I will try to do something. Okay, I will try to get these notes. I will see. Lymphocyte. Yes, lymphocyte is a particular cell which is doing specific immunity. Specific immunity means, suppose there is a particular bacteria for TB. Okay, and there is a particular bacteria for typhoid. So what is lymphocyte doing? Lymphocyte is basically giving you specific immunity in a way that the particular lymphocyte who is designed to have TB bacteria, to kill TB bacteria, they will be killing TB bacteria, but never typhoid bacteria, never plague bacteria, never the virus from any other sources. They will be only and only designed to kill TB bacteria. So they are giving you a specific immunity. Okay, specific immunity means what? It is very, very specific in its mode of action. That is what is a lymphocyte. That's called as immune system. Immune system is always specific in nature. Rest all WBCs, they give you defense. But lymphocyte gives you specific immunity. It's a part of defense, but very, very precise. Okay, fine. So what is it, the difference? Let's see. WBC gives you defense. It helps in defense. But when you talk about lymphocytes, lymphocytes give you what? Lymphocytes give you, where is lymphocyte? Here. It is giving you specific immunity. Okay? Fine. So let me wrap up the class for today. Just go back home and study. Okay? Fine. Let us go back home and study. And please study the test part also, and don't bunk your test. It is very, very important. Otherwise, how would we be able to have more of things from your side? I need to know more of things from your side also. And please don't do negative markings in test. They are going to be very, very expensive in long run. So go back home, study. If you have any doubts, you can ask me on the WhatsApp group also. Okay, fine. Thank you so much from my side also. Okay. Bye-bye. See you soon. Take care. Good night.